9点了才落山哦。
对对，我们已经到了东部时区的最西边了。
对，跟
夏天
Michigan 差不多
8月，
可能。
对对对。那 Michigan 是中部时间吗？也是东部时间。
东部时
Michigan 是东部时间，
哦，OK OK，那差不多，那差不多。对对
十
对。
二小时，差12小时。
嗯，我挺喜欢这个 long day 的。
但是你现在已经 recruit 了多少人了？
哎，4、5个人吧。
哦，
不是，
嗯，
有2个 technician，对，然后2个 PhD 学生，1个 postdoc，对。
嗯，
首先，要不你试一下你的 PowerPoint。
啊，我试一下，对对，我试一下看看
对
能不能跟我
对对对，是。嗯，
嗯，
哦，他
这是
让我退出一下，我要先退出一下。
OK。
哇，小伟，那以后可以经常见面了啊。
对对对对对。
Nice。
对，国内这几天放假的话，大家好像也都没有去 travel，学校里的人。哎，slides 可以了。
这2年中国的那个好文章，那个爆发式增长的，那个
对，就说现在卷的不得了了，已经。
哇，这个做了250期了，这么快？
对，今天是第258期。嗯，第一期开始应该是在2020年吧，现在已
对，
经
第一期是麒麟，
算
第一期麒
现在
麟讲座。
可以啊，没问题啊，没问题啊。
没问题。
没问题没问
嗯，
题。
好好，我这个 Zoom meeting 这个电脑上没装，刚刚装的。
哎，现
哦，
在国内用 Zoom 好用吗？就是有
就是开会没什么问题啊，还好。嗯，
啊，
但是我今天特地升级了一下，它有的时候不升级就会卡，就
嗯
用之前先升级一下就行了。
嗯
哎，Jandy，现在美国大学里面主要还是用 Zoom 是吧？
基本都是 Zoom，对，NIH 也是用 Zoom。
哦，我
嗯，
们有时候跟国外的那个，有一些 institute 吧，他们就一定要用 Microsoft Teams，都不能用
有，
Zoom。
对，两个都有，就 Team 也有用，但是就说一般就一个学校就是要么 Zoom，要么 Team，我觉得就是这个比较 dominant 的这个 supplier。
哦，
最近我
对对，
们这几次跟 PO 聊天的话，一般都是Teams，他
嗯，
们好像 prefer 那个东西，
对，
但是
好像
换来换去
公司
挺烦的。
private sector 一般好像 Team 用的比较多
Teams
嗯，
叫 Team
，Team
嘛？
就 Microsoft 的那个
这
Team。
微软的
哦
一个，
哦哦，
我们这也
我
要求用 Teams。
哦，
对，不知道为什么现在，对，有一个说法说是 Zoom，它这个好像比较 top 的几位也是华裔，这当然都是，也
嗯，
差不多时间到了啊，我们就开始了。 So hello everyone, welcome to the Metabolic Edge Talk Series 258. Today we are honored to have Dr. Zhuoxian Meng joining us. Dr. Zhuoxian Meng is a Qiushi Distinguished Professor at Zhejiang University School of Medicine. He completed his PhD training in Dr. Xiao Han's laboratory at Nanjing Medical University, and then received his postdoctoral training and served as a research investigator in Dr. Jiandie Lin's laboratory at the University of Michigan. Dr. Meng's research focuses on transcriptional and epigenetic regulation of energy metabolism, with an emphasis on pancreatic beta cell function, skeletal muscle-derived endocrine signaling, and immunometabolic crosstalks in obesity and type 2 diabetes. His work has identified key chromatin remodeling checkpoints and signaling pathways that govern metabolic homeostasis and disease progression. He has published over 70 peer-reviewed papers in leading journals, including Nature Medicine, Cell Metabolism, Molecular Cell, Developmental Cell, Journal of Clinical Investigation, and Journal of Experimental Medicine. He is a recipient of the National Science Fund for Distinguished Young Scholars in China. His topic today is chromatin remodeling checkpoints of beta cell dysfunction in type 2 diabetes. We have four panelists who will join in the discussion after the talk. Dr. Jiandie Lin and Dr. Yujing Chen from the University of Michigan, Dr. Zongwei from Mayo Clinic, and Dr. Zhenqi Zhou from University of California, Los Angeles. Meanwhile, our audience can post your questions in the chat and we will call you after the panel discussion. Okay, Josie, you can start your presentation now.
All right. Okay. Okay. Can you guys hear me clear? Oh, all right. It's okay.
Yeah, everything looks good.
Slides? Okay. All right. Okay. Thanks, Chunmei, for the introduction. Thank Xiaoxu and Chunmei for the invitation. It's my great honor to present my work. And very happy to see all my colleagues, all you guys today. And it's actually a holiday in China. I still really appreciate you guys come to join this seminar. This is actually 258. It's actually a great honor to introduce my work. So today I'm going to talk about our work on beta cell dysfunction, mainly focusing on protein remodeling. So we know that both genetic and environmental factors are very important for the development of diabetes. Over the past 2 to 3 decades, many loci, more than 300 loci, have been identified that are involved in the regulation beta cell in beta type 2 diabetes. Among all these loci, more than 80% are actually in the non-coding region, and most of these loci are actually associated with diabetes, with islet dysfunction. So then the question is why this mutation in the non-coding region play an important role in the diabetes is not very clear. So our lab have been focusing on how the environmental factor, including particularly in type 2 diabetes, including overnutrition, less exercise, or inflammation, integrate in how the environmental factor can be sensed by the body to control the gene expression so that to control the progression of diabetes. So we're focusing on the epigenetic mechanism, so we know that epigenetic have DNA methylation, histone modification, but our lab mainly focusing on the chromatin remodeling. So Um, our DNA, we know that our DNA is more than 1 meter long. It has to be compacted by more than 5,000 fold in order to form this kind of chromatin structure, which is very important for the DNA stability. But one gene needs to be transcribed. This very contains DNA structure need to be open. In this case, the chromatin remodeling complex will be kicking to regulate the chromatin accessibility so that to let the transcriptional initial complex to come in to regulate gene transcription. So chromatin remodeling complex mainly have four isoforms. The Swiss sniff is the largest isoform, and also other isoform. So different isoform actually play a different role in the organization of chromatin structure. For example, the I3 and CHD complex is involved in the regulation of nucleosome assembly. And the Swiss sniff is mainly involved in the regulation of chromatin accessibility. And also other isoforms, for example, INO80 is important for the regulation of the chromosome editing. So my lab have been mainly focusing on since I was working in JD Lin's lab, we mainly focusing on the Swiss sniff complex. So over the decades, people have been the Swiss sniff complex have been further classified, divided into three different sub-isoforms, including BAF, the canonical BAF, and also PBAF, means polybromo associated BAF, and also non-canonical BAF. In the PBAF, there's a very famous component called BR7. In the non-canonical BAF, there's a component called BRD9. So in this audience, Dr. Wei Zhong actually in 2018, they actually make a very interesting discovery, find that vitamin D actually.
Hey, Zhouxian, you are freezing now.
说到重要的 points 的时候，这个网络都不能承载了。伟聪的 paper，
Maybe I should continue.
我要把这张图截个屏，这张 slide。
有可能跟他打个微信电话，他
哦，
不一
我来，
定知道。
我来，我来，我来。
对，
对他别不知道，我来，
他不知道，他照样在讲课。
现在可以了吗？喂，
可以可以。
现在能
可以了
听到了。
我们学校有两个网络，有一个网络是那个 secure 那个网络，好像可能是不是因为 Zoom meeting 可能不是那个，后来我换成另外一个网络，现在 OK 了啊。
可以了，
现在没问题。
那个 secure 那个网络不太行，那个可能是有一点点 block 了啊。
讲到维他命 D。
好的，OK。刚才我听到说这个 freeze 了，OK，all right。Okay, so Dr. Wei Zhong actually in 2018 they published this very nice work. Actually, they found that vitamin B can switch the PBAF and the canonical BAF, non-canonical BAF to regulate the beta cell survival and play an important role in the control of glucose homeostasis. So my lab has been mainly focused on the the canonical three-leaf complex, which is made up by 18 protein, up to 15 components. One gene needs to be transcribed. Transcription factor will bind to the promoter, and in this case, the three-leaf complex will be recruited through the BAF60 component, which contain in mammal contain A, B, C, three isoforms. And in our past, from 2009 to now, in the past 17 years, we actually find that different isoform of BAFCC actually have different regulatory mechanism and distribution, thereby recruit different complex to regulate downstream gene expression and metabolism. And we actually find that BAFC is particularly high in white muscle and very important to the control of white muscle formation and glucose sensing and homeostasis. And in addition, we also find that BAFA is important for the control of red muscle function. And today I'm going to share our work actually on the BAFCC involved in the regulation of beta cell function. And we actually find that BAFA is important for the control of beta cell itself identity and function. And the BAFC, we found that instead of directly regulate beta cell function, is actually regulate the control the regulation interaction between beta cell and macrophage, thereby play an important role in the control of islet immune function, beta cell insulin secretion. And surprisingly, we actually find that BAFB not directly regulate the beta cell function, but regulate SNI homeostasis. I'm going to talk a little bit about BAFB in the final slides. During the study, we also find BAF also important, pretty important role in the regulation of liver lipid metabolism. For example, in collaboration with Dr. Eugene here, also in the audience, we actually find that BAF-A is important for the control of cholesterol homeostasis, thereby control the progression of atherosclerosis. Recently, in our lab, we also find that BAF-B particular is important for the triglyceride sensing and fatty liver disease. And in addition to the regulation of energy metabolism, we also find that BAF-A and C can also regulate many muscle secreted factor we call myokines or exokines. Actually, they are either working through paracrine to control the muscle regeneration or working through the endocrine to either control islet function or other tissue, for example, liver and fat metabolism. Today, our main focusing is islet. We know that islet is located in the pancreas, which is only constitute about 1 to 2% of the pancreas volume. But the islet actually have a lot of blood vessel, receive 10 to 15% of blood vessel in the pancreas. It's a cluster of 1,000 to 2,000 cells in the pancreas. Recently, through single-cell sequencing analysis, people actually find that in islet not only contain the insulin secrete cell, beta cell, and the glucagon secrete alpha cell, and other hormone secrete cell, but also in islet it also have other cells, for example, endothelial cell and macrophage T cells. Recently, in a study from Dr. Liu Yong's lab also find that the endothelial cell in the islet is also very important in the control of islet homeostasis and beta cell insulin secretion. So when the beta cell, when the glucose increase in the circulation, beta cell will sense the glucose so that to promote the insulin secretion through the signaling pathway and eventually calcium signaling and finally leading to the docking of the insulin granule to secrete insulin to the blood vessel. One thing I want to mention that actually in our body, our beta cell have two phases of insulin secretion, the first phase and the second phase. A very acute first phase within minutes and followed by 1 to 2 hours of second phase of insulin secretion. during the progression of type 2 diabetes, the at very beginning, the first phase of insulin secretion will disappear, and in the meantime, the second phase will compensate to increase. And toward the end of diabetes, both first phase and second phase of insulin secretion will be decreased. Eventually, type 2 diabetes will also need to do the insulin treatment injection so that to maintain the glucose in a certain range, normal range. In 2018-19, a very famous study from the Taylor's group, so-called direct study, they actually found that very through the lifestyle intervention that can reduce body weight 10% can lead to the remission of type 2 diabetes. And very interesting, they find that about 50% they have the response, they can lead to remission, but the other 50% cannot achieve remission of diabetes. And the determine is whether the first phase of insulin secretion can be restored by this lifestyle intervention, exercise, or dietary intervention, suggesting that beta cell insulin secretion, particular the first phase insulin secretion, is very important for the progression of type 2 diabetes and therapeutic outcome. So in order to really look at how the beta cell responds to glucose, we actually develop this perfusion system to help us to dynamically, very accurately to evaluate the first phase and second phase insulin secretion. This actually show a picture showing us that during the progression of diabetes, at the very beginning, first phase and second phase of insulin secretion increase upon high-fat diet feeding, and gradually at 3 months it still increase, but toward the 5 to 6 months, both first phase and second phase insulin secretion is decreased. So using this device, we can actually monitor the dynamic adaptation of insulin secretion during the disease progression. And this device have been used by many labs in China, and also some lab in the United States. We actually collaborate, published more than 10 papers using this device to really map out the insulin secretion function. And also using this system, we actually this paper has already been published actually to really elucidate dynamic adaptation of insulin secretion, and also find that dietary intervention can work through CTCF, which is a factor that can regulate the chromatin structure, so that to regulate the gene expression that are important for insulin glucose uptake and insulin secretion in beta cell, so that to restore the beta cell function in diabetes. This is published in 2022 in JN as a cover story. At the beginning, I mentioned that in diabetes, people have identified more than 700 loci associated with diabetes. And the majority, 90%, are actually located in the non-coding region. Then the question is why this non-coding region, how this non-coding region mutation can contribute to the progression of beta cell dysfunction in diabetes. Recently, by using the ATAC sequencing, even in single cell or in both mice and humans, people actually find that type 2 diabetes beta cell dysfunction is associated with global selective chromatin remodeling accessibility change. But how this chromatin remodeling is regulated is not known. We try to identify upstream regulator that are important for the control of the chromatin structure, thereby contribute to the progression of diabetes. So in order to address this question, we perform ATAC sequencing analysis on different diabetes model, including high-fat diet, OB, and DB. In this case, we show OB and DB here. We can see several, more than 1,000 of peaks are actually shared, open, and closed in diabetic condition in islets. And these shear and open and closed peaks are associated with genes that are important for hormone secretion, calcium transport, and also other immune regulation. And if we look into these peaks, we actually find that these peaks actually are not located in the promoter region. It's mainly located in the distal enhancer region, which is also consistent with Dr. Wei Zhong's lab. They actually find that with vitamin D actually can also regulate the enhancer region instead of uh, um, uh, the promoter region. But how this distal enhancer region contribute to the progression of diabetes is not clear. And also, in our normal transmission regulation study, we mainly focus on the promoter. The enhancer actually is quite, particular distal enhancer is quite far away from the gene body. That's why it's very difficult to really study all these mutation, human mutation involved in the development of beta cell dysfunction in diabetes. Then we want to know whether this upstream regulator involved in the remodeling of the chromatin. We performed the ion sequencing analysis and found that indeed many genes are shared downregulated or upregulated in beta cell in diabetes. For those upregulated, it's known that many inflammatory pathway which lead to either inflammation in diabetes is shown. But also in the downregulated gene, we find that gene involved insulin secretion, glucose metabolism is downregulated. We also identify 14 genes that are involved in the chromatin structure. Chromatin remodeling is also downregulated. And then we perform a small SI knockdown experiment to look at whether this downregulated gene is involved in the regulation of insulin secretion. Very interestingly, we find that some genes, when they are knocked down, they have no effect on insulin secretion. Particularly interesting, BRG1, which is ATPase in the three-sleeve complex, is why BRG1 is knocked down has no effect on insulin secretion. And we found that smart CD1, which include BFA, knockdown has a very most robust effect on insulin secretion. And BRD8 is also another homolog as BRD7 and 9 is also shown downregulation of insulin secretion. And this work actually is also found in Dr. Li Yan's lab from Case Western University. They actually find that BRD through screen, they also identify BRD8 knockdown can regulate insulin secretion. They published a paper in Cell Report in 2019. And more interesting, we found that BAFA can dose dependently regulate insulin secretion. When you knock down BAFA from the 20% to 40, 60, 50%, is dose dependent regulate insulin secretion, suggesting that BAFA level in beta cell is indeed important, maybe for insulin secretion. And through contact sequencing analysis, we can globally look at BAFA occupied in the genome. And we found that for those shear differential accessible peaks, about 40% is actually occupied by BAFA, suggesting that this single molecule maybe is very important for the chromatin regulation accessibility change in diabetic condition, and maybe play an important role in the so-called, at the beginning we call selective chromatin remodeling of beta cell dysfunction. Indeed, we actually, at both mRNA and the protein level, we found that BAFA is downregulated in high-fat OB/DB mice, and both mRNA protein, as you can see, the beta cell BAFA protein level is downregulated. And also in collaboration with doctor in the hospital, we actually find that BAFA is also downregulated in the beta cell from type 2 diabetes, suggesting that BAFA may be important for beta cell dysfunction in type 2 diabetes. Then we generate a beta cell specific knockout BAFA knockout mice. We actually use multiple different crate. I would like to thank Jandy for help me to order this. Some of the crate mice actually is ordered by Jandy. When I come back to China, I asked whether I can buy some mice for my beta cell study in my in China. He actually helped me a lot on this project. And we using great ERT mice, we actually can knock out BAFA in the adult mice to really look at the beta cell BAFA function in the adult mice. We also use other crate, for example, PDS1 crate can delete BAFA in at the very beginning. We actually find that BAFA is important for the control of beta cell identity. I'm not going to talk about that because of time limitation. When we knock out a BAFA in the adult mice using the MIP-crate ERT, we can see the fasting glucose tolerance are all elevated, and glucose tolerance is impaired, but insulin tolerance is not affected. And we also find that using our perfusion system and whole eyelid calcium imaging, we found that both calcium response and first phase and second phase insulin secretion, and also maximum insulin secretion in response to potassium chloride, all decrease in by BAFA knockout in the adult mice. Since BAFA is decreasing the diabetic condition, then we wonder whether if we overexpress BAFA, whether you can restore beta cell function and glucose homeostasis. Indeed, we found that expression of BAFA and decrease can restore the decrease the blood sugar level and also improve glucose tolerance and insulin tolerance. And we also find that the insulin secretion and calcium response is also largely restored in DBDB mice by BAFA overexpression. But in the clinical, normally we cannot do the pancreatic ductal infusion, right? But we can use.
看上去又掉线了，
Something wrong? Oh,
掉线了。
祖先你好像 mute 了，
好像他过一段时间就会提醒我，让我退出来，不知道为啥，可能 Zoom 在国内不知道是不是有点问题。Anyways，好，现在可以了啊。
没问题的。
啊。Then we found that because in the clinical we cannot use the pancreatic ductal injection. We actually in the clinical we can do the islet transplantation. We wonder whether because during the transplantation we noticed that a lot of the human islets actually are not functional very well. We wonder whether we can treat the islet from human. In this case, we use both mouse islet and human islet and treat with either GFP control or BAFA virus to see whether it can improve beta cell function after transplantation. Indeed, we found that AV treated BAFA treated islet can improve beta cell function and glucose homeostasis. And then we wonder whether BAFA deficiency can contribute to the alteration of chromatin accessibility in beta cell. Indeed, we find that chromatin structure is altered, and also the altered chromatin accessibility is associated with gene involved in insulin secretion. And because BAFA is a cofactor, it has to interact with transcription factor that can regulate the chromatin structure. A long story short, we actually identify NKS 6.1 can interact with BAFA to regulate the insulin secretion. And then we want whether our finding have any clinical relevance, collaboration with Hu Cheng from Shanghai 6 Hospital by analyzing more than 10,000 human samples. We actually identify a mutation in at 278 amino acid. Is a valine to methionine mutation, and this mutation will cause a structure change of BAFA. And this mutation is actually very conserved between different species. And this mutation will cause the downregulation of both early phase and the later phase of insulin secretion. Then we generate these mice contain this mutation, and we perform the metabolic study, and we found that this mutation can accelerate high-fat diet-induced glucose intolerance and induce beta cell dysfunction. And also, very interesting, we found that BAFA mutation or deficiency will lead to the downregulation of GLP-1 receptor and GIP receptor. We know that GLP receptor actually recently very hot, popular in the field that can reduce body weight and also improve energy metabolism in the humans. And one of the actually at the beginning, the important function of GLP and GIP receptor is actually to promote the insulin secretion, and we find that BAFA deficiency can attenuate the semaglutide induced upregulation of insulin secretion, suggesting that BAFA may be an important contribute to the therapeutic benefit of GLP or GIP receptor agonist. In summary, so this part, we actually identify BAFA is a very important chromatin remodeling regulator that can interact with transcription factor NK6.1 to regulate genes that are important for insulin secretion. But in human, in type 2 diabetes, BAFA will be decreased. In human who contain the mutation, I want to mention that this mutation is only happening in Asian, major happening in Asian population, but not in European. This mutation will cause the dissociation of BAFA from the NKS 6.1, thereby lead to beta cell dysfunction. So I want to switch the gear to the next part. We know that in islet not contain beta cell, but also other cell type, for example, immune cell. And among the immune cell, about 70 to 80%, 60 to 70% is macrophage. This TM image is showing us the interaction between beta cell and the macrophage. This is actually a very nice review, right, by Fu Wenxian, our CADA member. They actually write this review, summarize the non-function regulation of macrophage and beta cell. And it's known that macrophage has secreted many cytokines to impair beta cell function. And then we want to ask whether in the progression of diabetes, whether beta cell can also actively involved in the regulation of beta islet homeostasis and beta cell function, whether it can actively regulate macrophage activation as well. And at the beginning, I mentioned that BAFC is very important for the control of glucose sensing, and because both muscle and beta cell, they are very active sensing glucose and control either glucose uptake or insulin secretion. And we're in our during the study, we also Dr. Eugene and Jifeng from University of Michigan, also in the audience. They actually also in the blood vessel smooth muscle system. They actually find that BAFA and C play important role in the smooth muscle homeostasis through either directly regulate the smooth muscle survival or regulate the immune microenvironment, thereby play an important role in the development of cardiovascular disease. And we also find BAF involved in the regulation of adipose tissue homeostasis and liver function. I already talked about this earlier. And then we want to ask, since BAFC is important for the control of glucose homeostasis, we wonder whether BAFC also important for the control of beta cell glucose sensing and the GSS function. Indeed, we found that BAFC, similar to BAFA, is also decreased in diabetic beta cell. This is in different mouse model, and also in humans, BAFC is decreased in beta cells. And Then, in order to look at how BAFF involved in the beta cell function, we perform this bio-ID analysis to map out the interactome between BAFF and other protein. We consistent with our hypothesis. We found that BAFF can indeed interact with 3-SNF complex. But we very interestingly, we found that the top geo term is actually the regulation of RNA homeostasis. And among these 70 proteins, we found that many proteins are involved in the RNA metabolic process, RNA splicing, mRNA processing, and also many proteins are actually located in the ribosome and involving ribosome RNA homeostasis. And some protein, including MPL1, which is typical nucleolide protein involved in ribosome biogenesis. And then we wonder why normally BAFC should be located in the nuclear plasma. Why is interact with protein in the nucleolide? It's not clear. We know that in our cell, nuclear is very important for the gene transcription. But also in the cell, we also in the nuclear, we also have a nucleolide structure, which is very famous for its ribosome biogenesis and thereby contribute to the gene protein expression. Then we want to know why BAFC can interact with protein in the nucleolase, right? Whether it's involved in the regulation of nucleolide homeostasis and RNA processing. And recent study actually showing that nucleolide not only participate in the ribosome biogenesis, but also serve an important hub in the control of cell stress response. Then we want to know whether in beta cell dysfunction or whether it's also associated with nucleolide stress. And this is a TM study. We actually find that in all different kind of diabetic mouse model, we find that a nucleolide volume is actually increased. And also in human islets, we treat the islet with the palmitic acid and the TNF-alpha, which mimic the inflammatory and lipid toxicity situation in diabetes. We also find that PT can induce enlarge of nucleolide volume. And later on, in human pancreas section, using fibrillarium to look at the nucleolide size, we actually find that nucleolide volume is indeed increased in type 2 diabetic beta cells. And furthermore, we actually are using cell culture to find that the metabolic stress using the palmitic acid and the cytokines cocktail can induce the enlargement of nucleolide. And also, MPL1 is a known protein normally located in the nucleolide. Why is translocated from the nucleolide to the nuclear plasma is considered as a hallmark of nucleolide stress, suggesting that beta cell dysfunction in type 2 diabetes is involved associated with nucleolide stress. Then we want to ask whether BAFC may have some role in this response. We generated BAFC specific beta cell knockout mice, beta cell specific BAFC knockout mice, and different from BAFC. When you in normal child diabetic condition, BAFC beta cell knockout mice have no phenotype. But when we put the mice on the metabolic stress condition, high fat diet, you can see although the body weight no difference, but blood sugar is increased, TGT is impaired, and the insulin secretion, both first phase and second phase, also impaired. And very interesting, we also found that the nucleolide volume size is increased by BAFC knockout. When we look at the transcriptional gene program, we are consistent with impaired insulin secretion. We found that the gene involved in insulin expression and secretion is decreased, which is consistent with downregulation of insulin secretion. But also for a lot of genes you can say are upregulated. For those upregulated genes, there are many involved in inflammatory pathways. If we look at all these inflammatory genes, you can see the chemokine CCL5, CXCL9 and 10 are increased. And in the meantime, several gene red family protein is decreased. And red protein, at the beginning, we don't know what is a red protein. It's actually normally expressed in two sites. One is expressed in the gut, in the intestine, which is play important role in the control of gut intestine immune environment. It's antibiotic, antimicrobial peptide, and also RAG, so-called regenerating islet derived protein, is also expressed in the pancreas, and during the development is important for the regeneration of islet. And why is expressed in the beta cell in the islet is not known. And we also find that among all these red protein, Reg3b is the highest, and its mRNA protein is decreased in biopsy knockout. Then we want to find, as we don't said at the beginning, people don't know why RAG3B, RAG family is expressed in beta cell. What's the function of RAG? Then we want to know what's the role of RAG in the vaccine deficiency regulation of islet homeostasis and beta cell function. We purify the RAG3B protein and treat the beta cell. We found that beta cell either proliferation or insulin secretion under both normal condition and the stress condition are not affected by RAG3B treatment. Then we treat the macrophage with LPS with or without RAG3B, because LPS is normally high in the diabetic condition. In this case, we mimic the gut leaking and the toxin condition in diabetes. We found that the LPS induced elevation of cytokines is a significant decrease by RAG3B pretreatment, and the impaired insulin secretion is also restored, suggesting that RAG3B may work through macrophage, thereby regulate the islet immune microenvironment and contribute to the regulation of beta cell insulin secretion. And later on, we do several studies to prove that RAG3B downregulation is involved in the BAFC deficiency in the beta cell and contribute to the islet homeostasis and beta cell insulin secretion. In this case, we either treat through IP injection of RAG3 protein. Then and also we do the pancreatic ductal infusion of RAG3B AAV. We can find that the impaired glucose tolerance, decreased calcium response, and insulin secretion in beta cell, BAFC deficiency beta cell is largely restored by RAG3B IP injection. And the transcriptional analysis also suggesting that the downregulation of insulin secretion related pathway is, and also the upregulation of the inflammation is restored by RAG3B IP injection. And because in diabetes condition, the RAG3B is actually decreased. This is control. This diabetes RAG3B is decreased. We also using the pancreatic ductal infusion of RAG3B under the RIP promoter allow us to express RAG3B only in beta cell. We can see the RAG3B can be restored to the normal level as control, and it can actually very nicely reduce the blood sugar level in diabetic condition and associated with improvement of insulin secretion and calcium response, suggesting that RAG3B is sufficient to restore islet inflammation and beta cell function. And then because we work on the protein remodeling, normally the next step is we try to find a transcription factor that can interact with BAFC to regulate the RAG3B. We perform the ATAC sequencing analysis and find that indeed gene involved in insulin secretion, the accessibility is compromised, is regulated. But when we look at the RAG3B gene loci, although the mRNA level is decreased in the knockout mice, but the chromatin accessibility is generally no affected. There's no opening or close surrounding the RAP3B gene family, which is really a trouble for us because we have no clue. Uh, if normally we can find some opening and close, we can do the motif analysis and find transcription factor. But in this case, we cannot find the protein that can interact with BFC, which this project stuck there for a very long time, at least 2 to 3 years. We have no answer. But in the end, we find that through many different kind of trial, in the end, we find that BFC deficiency actually does not regulate the transcription of Rec3B. It's actually regulate the MI stability. You can see in the BFC knockout mice, Rec3B MI stability is decreased. And but in other for other genes, for example, insulin PDS1, the transcription level rate is decreased. But the Rec3B transcription itself is not affected. And we also confirm this result using the cell culture, find that knockdown biopsy lead to decrease of Rec3B mRNA. We actually also perform IP experiment, find that Rec3B can pull down biopsy. On the other hand, biopsy can also pull down Rec3B. But BFC is a cofactor. It cannot interact with Rec3B. It's mRNA. Right? Then we perform several studies, including the prediction using RBP suit prediction can predict the Rec3B RNA binding protein. We're also using pull down the Rec3B RNA binding protein and perform the mass spec analysis. And in combination with the BFC bioID, and eventually we found that NPM1 is a linker between BFC and the REC3B mRNA. And in contrast to BFC, NPM1 actually can promote a degradation of REC3B, suggesting that NPM1 may be the linker. Later on, we performed a very nice in vitro study by this student, very talented student in my lab, who right now is at the University of Oxford for pursue his PhD study. We actually found that NPM1 can direct interact with REC3B, and only in the presence of NPM1 and the BFC can interact with REC3B. And we'll also map out the interaction domain on BFC protein or REC3B mRNA that can mediate interaction with NPM1 so that form this RNA protein complex. And also we as required by the review, we also perform the in vivo interaction experiment using the either proximity ligation assay to look at the NPM1 BFC can indeed interact with each other. And we performed also the IF study, immunofluorescence, to look at the protein actually can indeed interact with each other. And also we performed the RNA scope and find that BFC can indeed interact with Rec3B in normal condition, but not in the knockout. And in the normal condition, NPM1 is not normally located in the nucleoli, cannot interact with Rec3B. But in the knockout, NPM1 will be translocated from the nucleoli to the nuclear plasma so that it can interact with Rec3B to promote degradation of Rec3B. And then we want to ask whether, on the other hand, if we restore BFC deficiency or we active upregulate BFC expression, whether it has opposite effects, particular to look at whether BFC KM function is sufficient to alleviate the nucleolar stress. Indeed, we found that BFC knocking overexpression can reduce the nucleolar volume in response to hyperdiaphyting associated with improvement of insulin secretion. And after this study, we want to know whether we have any solution, any way to manipulate this pathway to improve beta cell function. We performed this exercise study So we put the mice on high-fi diet, then let the mice run on the treadmill for 1 hour using the middle speed, like 10 to 14 meter per minute. And 1 hour later, we collect the protein islet for the protein and mRNA analysis. We actually find that both mRNA and protein of REC3B, BAFC and REC3B is upregulated. This is only a single exercise at 24 hours after exercise. And we train the mice for about 1 month. We can see that the insulin secretion is also restored, associated with improved beta cell functional gene and alleviation of islet inflammation. This is actually one exercise. This is after another set of mice after 1 month of intervention. So in summary, so we actually find in the beta cell. So there's a new mechanism involving BAFC and that can interact with REC3B mediated through the NPM1 protein. In normal conditions, REC3B will kind of preserve BAFC will preserve REC3B so that to maintain the islet immune environment inflammation, so that the beta cell function will be preserved. But during the diabetic condition, REC3B will be decreased, BAFC will be decreased, and the REC3B will be degraded by NPM1 translocated from the nucleolate to nuclear plasma, so that the islet macrophage will produce more cytokines to attack beta cell, cause beta cell dysfunction. And using exercise, we can actually restore this kind of immune, this pathway to promote the secretion of REC3B so that to maintain islet inflammation and beta cell function. And actually, this is actually modified from the review article from Wenxian. We actually different from the normal. We know the normal model is macrophage can secrete a lot of cytokines to attack beta cell. But in our case, we actually find that in beta cell, BAFC, NPM1, REC3B from this kind of regulatory machinery can regulate the REC3B through paracrine mechanism to maintain the macrophage activation and immune cytokine production so that to maintain the immune microenvironment beta cell function. And I also want to mention that study from Yushel's group, they actually find that insulin B 9-23 is also secreted by beta cell that can act on macrophage to regulate the macrophage inflammation and beta cell function. So I have three slides. Just want to mention that what's the role of BAFB in beta cell, right? And we actually cross the BAFB. I'm sorry, this is Cray, should be Cray here. We crossed the BAFB flux mice with PDS1 Cray, and we didn't see any effect on blood sugar, insulin secretion, and glucose homeostasis. And actually, this is actually inspired by Qilin, and originally from University of Michigan. Right now, it's in Virginia, and they're actually using the whole body ERT mice to look at one gene function. When we cross the BAFB with the CAG ERT to induce BAFB deficiency in the whole body, and then we found that. At the beginning, we used 100 milligram per kilo, and we found that mice die within 2 to 3 weeks after injection of tamoxifen. And which is really at the beginning, we don't know what's happening. But later on, we found that we knockout of BAFC can cause the totally destruction of acinar cells, and the structure of the pancreas is totally disrupted. If we inject a lower dose of tamoxifen, cause a less severe deficiency of BAFB, will cause less severe islet pancreatitis, and this suggesting that BAFB's deficiency will cause dose dependently cause the acinar cell destroy and pancreatitis in the mice. And then later on, we wonder whether this effect is only is in acinar cell. We actually create cross the mice with cellular, and also this is also inducible knockout. We can say BAFB is knockout is inactive. It's not expressed in the acinar cell. When it's consistent with whole body deficiency, you can see the knockout of BAFB in the acinar cell cause normal condition. You can see the pancreas has very nice structure and islet in the pancreas. After knockout of BAFB in the acinar cell, you can see the pancreas is totally disrupted. Okay, and the weight is decreased and associated with fibrosis, which is indicated by muscle staining here, a lot of collagen deposition. And we know that in the pancreas, the acinar cell, the exocrine acinar cell produce lipids and the proteins, which is important for the digestion and absorption of protein and lipids. It has a very extensive interaction with endocrine cells, this islet. And if the acinar cell exocrine have inflammation called pancreatitis, it actually can cause the inflammation and can gradually destroy the beta cell, lead to diabetes called type 3C diabetes. And we are still try to, because the type 3C diabetes, although it's not happen very high percentage in the human population, is only about 1 to 2%, 1%. But in the world, we have more than 5 million of people with type 3C diabetes. So we are still working on the hypothesis that maybe biopsy may play an important role in the type 3C diabetes through moderating acinar cell homeostasis and pancreatitis. All right. Sorry for the internet. I would like to thank all the people who some are already graduate. Xingyuan, Ruoran, they actually Wu Qingqian, they actually play an important role, make great contribution to the BFA project, and also Zhou and Chen An and also other students, and also Shuai Shuai, they contribute a lot to the BFC in beta cell. And Yuting and Zhou also made a great contribution to the beta BAFB in acinar cell and inflammation, pancreatitis. I would like to thank also my PhD mentor, Dr. Han Xiao from Nanjing Medical University. I actually learned beta cell study, islet study from while I will pursue my PhD degree in Dr. Han's lab. And particularly, I want to thank my postdoc mentor, Dr. Jian Jianling. Really, I have been working in his lab for 8 years and really enjoy the study at Michigan. And I would like to also thank Luo Chen and Jing Li, who this Jing Li is not a Jing Li in China. It's actually from McGill University. Luo Chen actually worked with him as a postdoc, and they actually give us a lot of suggestions and the reaction on RAT3B protein. And a lot of other collaborators, I will not to list all the names because of time limitation. Thank all the collaborators. Thank you very much for your attention. And I would like to take any questions.
Okay, yeah, thanks, Zhexian, so much for this like a really insightful and inspiring presentation. So I think Zhexian really dug into how the chromatin remodeling regulated the beta cell pancreas functions through studying the BAF60, AEC, and B proteins. So next, we will move to the panel discussion part. So first, let's start with Dr. Jiandie Lin, please, from University of Michigan.
Yeah, well, really nice work, Zhuxian. It's really beautiful, and it's clear that you'll be more productive in Hangzhou than in Michigan. So we'll figure out why next time. So it's very interesting, the BAF knockout in beta cell